Today's topic is the neurotransmitters. Learning objectives are here. Neurotransmitters are the chemical substances and which carry the information through a synapse. They should have some criteria. They should be prepared presynaptically. They should have some mechanism for inactivation, and they need some stimulus to release its release at the nerve terminal. They should have some receptors on the postsynaptic membrane, and applying substance has biological effect and also antagonizing inhibit biological effects. Classification depending upon chemical nature. The 4 types of synapses, neurotransmitters. Depending upon function, they are excitatory, inhibitory. Example for inhibitory are given: presynaptic inhibition, postsynaptic inhibition, then social inhibition. Depending upon the chemical nature, amino group like GABA, glycine, glutamate, aspartate. Cholinergic group, acetylcholine. Monoamine group, norepinephrine, dopamine, serotonin. Neuropeptide group, endorphins, enkephalins, substance P, somatostatin, so on. Depending upon the chemical nature, as in the previous picture, they are mainly of 3 types: amino acid group and amines and others. Amino acid group, these are fast synaptic transmissions. They are inhibitory and excitatory like GABA. Amines, these are modified amino acids. The slow synaptic transmission will have both excitatory and inhibitory in action, and example noradrenaline. Under the group of others, acetylcholine and nitric acid will come. Excitatory neurotransmitters, example glutamate. In response to depolarization, some sodium channels open, awaiting sodium ions to enter into the cell. The membrane status to depolarize. If the threshold of excitation is reached, a sodium channel will open. These are because of the excitatory neurotransmitters. Here, inhibitory neurotransmitter prevents action potential in the postsynaptic neuron, like GABA is the most common inhibitory neurotransmitter, which is causing not the development of action potential, causes the hyperpolarization of the postsynaptic neuron. That means makes more negative, thereby inhibits the action potential. Receptors of neurotransmitters, they have several characteristic features. There may be types and subtypes of receptors for particular neurotransmitter. For example, epinephrine acts on alpha 1 and alpha 2, beta 1 and beta 2, and beta 3 receptors. They are located not only on the postsynaptic membrane but also may be present on the presynaptic membrane sometimes. These presynaptic receptors may decrease or increase the secretion of the neurotransmitters through the feedback action, feedback mechanism or feedback signals. The receptors are present in high concentration on the postsynaptic membrane close to these presynaptic areas which secrete the corresponding neurotransmitter. If a neurotransmitter remains in contact with the receptors for a prolonged period, the receptors are desensitized, that is, their action decreases. So the receptor molecules are phosphorylated, attached to the clathrin and endocytosed, so their number is reduced. This process is known as down regulation of the receptors. Conversely, if the neurotransmitter is deficient, the receptors increase in number. This process, the opposite action, is known as up regulation of the receptors. Here are the neurotransmitters and their function. The receptors for each neurotransmitter was given in the table with their action and with the related drugs and illness caused by the deficiency in the receptors. Now coming to reuptake, the neurotransmitters may be taken up from synaptic cleft into the presynaptic neuron. Active transporter system are involved in this process, as we all know that. One of such transporter system helps in the reuptake of the norepinephrine, dopamine, serotonin, GABA, glycine, choline, etc. The other transport system is concerned with the reuptake of glutamate. In contrast to these transporters, there are vesicular transport system present in the vesicles in presynaptic neuron. These help in the entry of neurotransmitter into secretory vesicles before their discharge into the synaptic cleft. The drug reserpine inhibits the vesicular monoamine transporters. This causes depletion of monoamines inside the secretory vesicle. So there is a dearth of the neurotransmitters at the synaptic cleft, which explains the antihypertensive action of the drug. Reuptake lowers the concentration of neurotransmitters at the synaptic cleft. So drugs inhibiting reuptake increases the concentration of neurotransmitter at the synapse. This is how the antidepressant drugs, which block the reuptake of serotonin selectively, produce their pharmacological effect due to excess serotonin at the synapse. Also, in ischemic and hypoxic conditions of the brain, reuptake of glutamate is inhibited, causes concentration of the glutamate increases at the synapse. That leads to high toxic effect of glutamate is exerted on the neurons, leading to their death. Now we briefly describe individual neurotransmitter. First we go with the acetylcholine. It is secreted preganglionic nerve endings of both synaptic, sympathetic and parasympathetic neurons at the postganglionic parasympathetic nerve endings, and also neuromuscular junction and CNS synapses. Its receptors principally are of two types, nicotinic and muscarinic. Nicotinic cholinergic receptors are formed by sub-5 units which encircle a certain ion channel. This channel remains closed in the inactive state. The composition of the subunits varies with the type and location of different receptors, but usually contains 2 alpha, beta, gamma and delta subunits. When acetylcholine becomes attached to the binding site present on alpha subunits of the receptor, the conformational change occurs resulting in opening of the channel allowing passage of sodium and other small ions. This results in the formation of EPSP, excitatory postsynaptic potential. As the case may be, and if threshold is reached, this will result in the formation of an action potential. You can also find some muscarinic receptors or different varieties. They are of 5 types, M1 to M5. They are sensitive receptors and act through G proteins. M1 type of receptor is present on brain, M2 found on heart, M4 is present on pancreas, mediating both exocrine and endocrine functions, while M3 and M5 mediate smooth muscular activity. Atropine blocks all the types of muscarinic receptors. The drug Prenzaphine is a selective M3 blocker, blocking receptors in oxyntic cells secreting HCl. Hence, it is used in the treatment of peptic ulcer. These are the functions. So far, I have explained in detail all of them. Now, coming to epinephrine and norepinephrine. So, these are called catecholamine neurotransmitters. Both are secreted from adrenal medulla, but only norepinephrine is secreted from sympathetic postganglionic nerve endings. Epinephrine secreted neurons are present in the brain. And both the norepinephrine and epinephrine act through alpha and beta receptors. Alpha receptors may be alpha 1 and alpha 2 types. Beta receptors are classified into beta 1 and beta 2 and beta 3 types. Norepinephrine has more pronounced effect on alpha receptors, while epinephrine has a predominant effect on beta receptors. Both alpha and beta receptors are serpentine receptors coupled with G protein. Here are some physiological actions of the epinephrine and norepinephrine given. Now coming to dopamine, it's another catecholamine neurotransmitter present in the synthetic junction at certain areas of brain. Dopamine receptors are mainly of 5 types, D1 to D5. Schizophrenia, the major psychosis is due to excessive activity of dopaminergic receptors. Until recently, D2 blockers like chlorphomazine were used to treat this disease, but concomitant blockage of D2 receptors in striatum lead to unwanted extrapyramidal effects. Newer atypical antipsychotics have minimal extrapyramidal side effects as they have less activity on striatal D2 receptors. Here we have some of the physiological actions of the dopamine. You can go through with them. Now serotonin, derived from the amino acid tryptophan, another important neurotransmitter, found in the platelets in the brain and the enterochromaffin cells of the main reflexes of the GIT. Serotonin has found numerous receptors. These are classified as a 5-HT1, 5-HT7, HT1 to HT7. Again, HT1, HT2 and HT5 groups are further subclassified. For example, 5-HT2 group contains 5-HT2A, 5-HT2B and 5-HT2C types of receptors. Newer drugs have been evaluated with action of specific 5-HT or serotonin receptors. For example, the drug sumatriptan relieves migraine by relieved by selective antagonist action of the 5-HT1D receptor. The drug andansetron relieves nausea and vomiting by antagonist action of the 5-HT3 receptor. The drug mosapride is a pro-kinetic, and it prevents gastroesophageal reflex by the selective antagonist action on the 5-HT4 receptors. These are some of the physiological actions of the serotonin. Now coming to histamine. Histamine is synthesized by decarboxylation of amino acid histidine as a neurotransmitter. It is distributed widely in brain and spinal cord, stomach, and pituitary gland. There are three types of receptors, H1, H2, and H3. H3 receptor is mainly presynaptic. Histamine is believed to mediate a wide range of CNS function, such as regulation of blood pressure, water intake, and parent-assured sexual behavior, etc. H1 antagonist, in addition to antihistaminic action, produces sedation and weight gain on chronic use. The drowsiness produced by conventional anti-allergic drugs are known to all. You can see some physiological actions of the histamine. Okay, and the other amino group of the neurotransmitters, glutamate and aspartate. They are excitatory neurotransmitters, and glutamate is involved 75% all excitatory transmissions of the brain. It is of two types: metabolic, which is coupled with the G proteins, and inotropic, like nicotinic cholinergic receptors. These are ion channels. There are three types of inotropic receptors: kinase, AMP, and NDMA. NDMA, N-methyl deaspartate receptor. These are involved in long-term potential learning, memory, long-term potentiation. A high-frequency stimulation for a short duration facilitates transmission of impulse across the synapses for a prolonged period. NMDA receptors are abundant in the hippocampus. They have following special features. They require glycine for the activation, and Mgg plus ions, magnesium ions, to keep the channel blocked at resting membrane. Potentially even glutamate binds to it. However, the neuron is depolarized through a separate glutamate sensitive AMP receptor. The Mg plus block is removed. Now, calcium ions enter the large amounts into the neuron. This ultimately leads to an augmented activity of AMPA receptors. Migration of more AMPA receptors to prosynaptic membrane. Additionally, a tetragrammatic signal in the form of NO or arachidonate is believed to pass from the postsynaptic to the presynaptic neuron, increasing further release of neurotransmitter. All these events releases in long-term potentiation, which in turn is involved in the learning and memory. Here are given some of the physiological actions mentioned in the slide. Now coming to GABA and glycine. The GABA and glycine are very important inhibitory neurotransmitter. GABA, aminobutyric acid, is widely present in the brain. It is formed by decarboxylation of glutamate. There are three GABA receptors: GABA A, GABA B, GABA C. GABA AA and GABA A, GABA C, GABA B, GABA, and all are chloride channels formed by live subunits encircling a small pore like nicotinic cholinergic receptors. In contrast, GABA B is coupled to G proteins. Chloride influx through chloride channels leads to hyperpolarization of the postsynaptic neuron with formation of inhibitory postsynaptic potential. Benzodiazepine, a widely used drug with the anxiolytic and sedative action, binds to the alpha subunit of GABA A receptors and increases chloride conductance. Now the glycine, when it binds with the NMDA receptor, it is excitatory. However, it has also a direct inhibitory action mediated like GABA by chloride channel. Inhibitory action of glycine is proved by the increased muscle excitability and seizures produced by strychnine, a glycine antagonist. this is clearly about the GABA, so far what I have explained. And here are some of the physiological actions of the glycine. And finally, opioid peptides. The drug morphine binds to receptor in the brain and gastrointestinal tract. These receptors are called opioid receptors. Investigations were carried out with a view of finding out the body's own ligands for the morphine binding receptors or opioid receptors. As a result of several peptides have been discovered which are called collectively opioid peptides. These opioid peptides are enkephalins and endorphins and dynorphins. Enkephalins include metenkephalin, which contains methionine, and leuencephalin, which contains leucine. The opioid peptides are divided from three main precursors: pre-enkephalin, which gives rise to leuencephalin, metencephalin, octapeptide, and hepatopeptide. Pro-opiomelanocortin, which gives rise to beta endorphin and other endorphins. Prodynorphin, which gives rise to dynorphins and neoendorphins. The opioid peptides have three types of receptors, and they are delta and mu and K. They are coupled with the G proteins of GQ type and decrease cyclic AMP activity. The mu receptors on activation augments K exit from cells, leading to hyperpolarization, while the other two types of receptors on activation close calcium channels. All the three receptors on stimulation produce analgesia. Other notable effects include sedation, pupillary constriction, and constipation can be seen. And look at, you can also see the metabolism over here. And so far all of them have explained polypeptides as a neurotransmitter. These are also called neuropeptides, like CCK and substance P, or some of the example. Neuropeptides, and before to this, I would like to talk about some of the other substances apart from opioid peptides. Many other polypeptides cannot act as a neurotransmitter. Substance P and tachykinins, hypophysiotrophic hormones, somatostatin, vasopressin, oxytocin, cholecystokinin, neuropeptide Y are some of them, are name a few. Also purinergic compounds such as ATP, adenosine, ADP also act as neurotransmitters. Among them, and nitric oxide is one of the very important one, produced from endothelium of the blood vessels, which are dilated by the action, and hence it is known as endothelium derived relaxation factor. They are synthesized from arginine actually. So nitrogen oxide synthesized acting as a catalyst. It acts via guanyl cyclase with the cyclic GMP as the second messenger. As it is a gas, it diffuses across the synapse easily, acts directly on guanylyl cyclase. It is believed to mediate long-term potentiation, long-term depression. Carbon dioxide, like nitric oxide, is another probable gaseous neurotransmitter and acting through guanyl cyclase. There will be, apart from the neuropeptides, and we have some, and their role, nitric oxide role, I have already talked about it. There are some co-transporters, and these there may be instances in which more than one neurotransmitter present in one nerve ending, and they are secreted simultaneously in the synaptic cleft. Such neurotransmitters together are called co-transmitters. For example, some cholinergic neurons secrete acetylcholine and VIP, with some adrenergic neurotransmitters secrete neuropeptide Y and ATP in addition to the norepinephrine. Well, an unproven theory suggesting when the neuron discharges a lower frequency, there is a secretion of small molecule neurotransmitter, whereas high frequency discharge leads to secretion of the bigger one and a polypeptide co-transmitter. Although the exact function of the significance of the co-transmitter is not so clear so far, simultaneous secretion of VIP and potentiates action of acetylcholine. Similar secretion of neuropeptide Y facilitates action of the norepinephrine. These are some of the co-transmitters. Coming to exocytotoxicity. Look at the picture carefully. The meaning of the exocytotoxicity is pathological process by which neurons are damaged by or killed by the overactivity of the receptors for excitatory neurotransmitters, glutamate such as NDDA receptor and AMPA receptors is called excitotoxicity. We'll go with the now growth factors, family, these are neurotrophins. They circulate throughout the entire body and important for maintaining homeostasis, critical for survival of sensory neurons. Without it, these neurons undergo apoptosis and they cause axonal growth, axonal branching and bit of elongation of the axons. Look at this picture carefully and see that growth factors shown in green is required for the neurons in order to survive, as they are limited extracellular resources. Some neurons shown in blue may uptake a disappropriate shape of survival factors, leading to the eventual death of the neighboring neuron shown in the red. Functions of the neurotransmitters: they have short-term functions and control of feeding and metabolic activity, regulation of neuronal phenotype, regulation of cell proliferation or apoptosis. So many functions are executed by them, which have been given in the picture. Cytokines are a diverse group of non-antibody proteins released by cells that act as intercellular mediators, especially in immune processes. Look at that. The lymphocytes. Cytokines are produced by various macrophages, granulocytes, and mast cells, fibroblasts, epithelial cell, lymphocytes. And once cytokine stimulating the cell, releases cytokine producing cells are mentioned in the left side of the picture frame. They release cytokines. They have their own receptors and they carry some message, and that will be executed acting on the target cell. Now the target cell will execute the function. These are the cytokines. Neurotrophins are the neurotrophin family, include now growth factor and brain-derived neurotrophic factor, neurotrophin 3 and neurotrophin 4. See over there how the neurotrophin is binding with the receptor. Cell survives or apoptosis. It all depends on it. Neurotrophins are NGF, BDNF and pronociceptirol and NT3 antinociceptirol and NT4 proprioceptirol, which increases the activity. Pronociceptirol promotes motor neuron survival but lack of role in pain. And coming to the receptors of the neurotrophins, they will act on various receptors. These are neurotrophin receptors. They may be on the cell membrane, clearly mentioned. T TRK, P75NTR, and once these enters into the cell, they execute by forming of secondary messenger survival. They are required for survival, apoptosis, proliferation, differentiation, axonal growth, dendritic, and oborization and synapse formation, retrograde signaling, crosstalk with other receptors. Okay, overall, and I would like to show you the receptors of the neurotrophins in this picture with this.